So now we're going to do our challenge awards. We had Albert and Seth already did a really good job of giving us an initial breakdown of that, so I will spare you the rehash. However, I will begin speaking in an awkward, unusually formal way, so we can put this on YouTube later. So, esteemed colleagues and guests, I'm honored to welcome you to the awards ceremony for the first two phases of the Biomarkers of Aging Challenge. Today, we come together to celebrate innovation, collaboration, and the pursuit of scientific advancement in the field of aging research. The Biomarkers of Aging Challenge was conceived to accelerate the development of accurate and reliable biomarkers that can predict chronological age, mortality, and morbidity. These biomarkers are crucial for understanding the aging process and for paving the way for interventions that can improve health span and lifespan. So I'd like to start with the awards for the first phase, chronological age prediction. We tasked our participants with developing models to predict chronological age with unprecedented accuracy using epigenetic data. And I'd like to start by recognizing our third place winner, Jakob Träuble from Team Zeta Partition. Did I get that horribly wrong? And I'd like to now recognize our second place winner, Lucas Paulo de Lima Camillo. And our first place winner for the first phase of the challenge, Julian Reinhard. And now the surprise one, because these already were given earlier this year, is the second phase for longevity and mortality prediction challenge, where Alamar Biosciences were very kind enough to let us run 500 samples through their panel, their inflammation and CNS panel. And I'd like to give Peter Wong an opportunity to say a couple of words.
Great, thanks. Dane, Jesse, Molly, great meeting. So last year I joined for the first time in the Buck Institute. And I remember going up to Dane right after the meeting saying, I want to get involved. We've created a solution that really enables, our customers to detect disease early. So we wanted to give access to this community. And we're really excited. And we ran 500 samples in about a week, turned it around, gave it back to the team, and off they went. So really excited about what we have so far. And I think there's a lot more to unravel as we continue through this challenge. And we're fully on board as an organization. And our missions are completely aligned with what you guys are doing here at Biomarker for Aging. So this is awesome. Really great.
Thank you, Peter. Thank you very much. And now we will give the final award. The winner of the second phase of our challenge for $40,000 is Team Kauai, which is Jakob Träuble, Corbinian Träuble, and Raphael Lemmer. So to all our winners, congratulations on your outstanding achievements. Your hard work and dedication have contributed significantly to this field. Obviously, the numbers speak for themselves. It really is amazing what you've been able to accomplish. And I'd like to extend our deepest gratitude to all the participants as well. It really was absolutely a community effort, and we look forward to seeing you for phase 3 of our challenge, which launches July 1st next year. And then finally, I just really would quickly like to thank all the sponsors and partners for the challenge itself. There are many, many sponsors and many partners that are helping to make this initiative possible. But I'd like to give a quick shout out to Methuselah, to Rosenkrantz Foundation, Vola Foundation, Foxo, Alamar Biosciences again, XPRIZE HealthSpan, Quadrascope, and again, the many sponsors of the consortium as a whole, and of course, everyone who works so hard from within their labs to make this endeavor possible. It was really an absolute community effort. So thank you to everyone. And now let's give them a round of applause. Yeah. And we're going to take a quick picture. After we're going to do your 3 talks, and then we're going to do the big group picture afterwards. Yeah.
So okay. Vadim and Kiara and Andrea, if you're here, come up please for the picture. Dane, you too.
Yeah.
Now.
Oh, now? Okay, we'll do now.
Oh, I thought.
No, it's good. It's great. Yeah.
You too, Dane.
Yeah. And so now we'd like to give our winners an opportunity to share their work in a few short talks. So let's start with Julian. Would you like to come up and share your work?
Yes. Thank you very much. This is the... Great. Thanks a lot. Yes, I'm Julian Reinhard, or during competition times, Darth Banter. And as the phase 1 winner on the hard chronological age target, I want to give you my 3 takeaways that I had from the competition here. Let's go on. First, probably to the surprise of no one, batch effects are a major issue. What you see here is one pretty representative CpG site on 5 different data sets. And the samples here are colored by chronological age. And you see here at the top 2 data sets with very different age ranges, having pretty much the same beta value range. At the bottom, you see the opposite, very similar age ranges in 2 data sets and very different beta value ranges for the CpG, as mentioned, representative for a lot of aging-associated CpGs. So as mentioned, these batch effects are a major issue. I tried for several weeks to align this, normalize this, somehow get rid of this, also with published methods. What I settled on in the end, however, was to just take these semi-raw beta values and shove it into the machine learning model and let the machine learning model deal with it. To really use a relatively large model to make end-to-end implicit batch correction for me, while being aware what the actual prediction target is. I would have actually liked to go even further, do this not on the already processed beta values, but do this on the raw signal intensities from the IDAT file, and would be really interesting how that further could maybe improve our prediction performance. But simply, this is, of course, an even larger endeavor. So first takeaway, for really maximum performance, end-to-end models, I think, are the place to go, even if they are a little bit black boxy. Okay, next one. While you need a larger model to do this implicit batch correction, it doesn't mean you need a gigantic model. I started out with a simple linear regression, which you see here. Really one weight per feature, 8,000 features. And what then back then got me into the top 3 was already a relatively small, actually, correction, non-linear correction network, as I called it. 500,000 parameters, relatively simple feed-forward network, which is small for machine learning terms. Of course, 500,000 parameters sounds like a lot, but this runs and can be trained in a few hours on an office notebook. Still, performance was not enough really to go forward with that. What I did is I used the strength that this was a relatively efficient and fast model and built an ensemble of those models. I think I used 4 dozen, that was probably overkill. And I, sorry, can we go back? Yeah. And I basically varied the input data sets to make the model aware. Sometimes batches are there, sometimes batches are not there. And also the remaining samples, of course. And this really put me then on top and made me in the first phase of the competition. So second takeaway, it doesn't always have to be a large language model. This still runs in a few hours now on a small GPU, but is still for machine learning terms relatively small. And to the third takeaway here about the data set sizes. So I didn't use the largest data set in the competition, 9,000 samples, 22 data sets from GEO, not by a long shot. But of course, I still needed a few thousand samples and needed to compile them. And without the data there, this is really not possible. So data is not everything, but without data, everything is not here. For age, this is somewhat manageable. There is a lot of data because it's a relatively simple outcome. For others, like phase 2, for example, this was much more complicated. And then, of course, there were some data sets that existed also for more rare outcomes, but are relatively high barrier to entry. And if your data set, for example, is behind an IRB approval or is very hard to digest, this doesn't just take the participants or the model designers a longer time. It also just makes a lot of projects not happen. So the highest impact thing you really can do if you have the possibility is to release data sets and make them as low entry as possible. And with that, thank you. And let's hear from the second place.
Thank you very much. Lucas?
Okay, I do apologize, everyone. This might be a little bit of a rough presentation. I was given a 1 hour heads up. So I put some slides together, but I'm going to talk a little bit about the model that I created for the phase 1 of the competition. So as you might have heard, it's called CpGPT, so CpG pre-trained transformer. And sort of the assumption of foundation models is that they're able to do many different tasks quite well. So just as an example for methylation, a foundation model for methylation might be able to impute missing values in a given array. It might convert different arrays. It might do reference mapping. You can get some sort of sample level interpretation. But specifically for the challenge, you can also fine tune it to predict chronological age and mortality. So when it comes to the specifics of how it works, it tries to integrate 3 main types of information. So the first one is the actual epigenetic information from the beta values that you get. The second type of information is the actual sequence around the CpG site that is encoded using another DNA large language model. And the third type of information is the actual positional information from the genomic locations in the chromosomes. Putting all of this together, the model creates an embedding that represents the sample. So it's just a vector of numbers that represents the methylome. And using the DNA LLM embeddings, you can actually query this sample embedding to predict the beta value in theory in any given genomic location, not even necessarily one that has been shown in an Illumina array. So first things first, it seems to understand the human methylome quite well. This is just some example data from the Illumina 450K array and just doing imputation. And the performance is quite good. You know, the Pearson correlation is pretty much 0.99. And amazingly as well, if you fine tune it in some mammalian species, you can actually impute the methylation values for unseen mammalian species. So there's something fundamentally going on that the model understands about these different CpG interactions, that the assumption is it could be harnessed to better predict chronological age. So the approach was, you know, pre-train with 100K plus samples and then fine-tune on some data sets that actually had the age label. That seemed to work quite well. So on the leaderboard, that was about position 10 or so. So I just did it again and again and again until I reached a plateau. And for the phase 1 of the competition specifically, there was also another rule that if you were within 0.1 MAE from another competitor, the competitor with the fewer number of features would win. So I just changed a little bit my strategy. So I did pre-train more and more, but just reducing the number of features, achieving essentially what was a very similar performance. So that's how the model performed. And yeah, that's it. Thank you very much, everyone.
Thank you.
So hello, everyone. First, I'd like to thank the consortium for organizing this challenge, all the people behind who made this possible, Dane, Seth, and Albert, among others. And I'm going to present our approach for the second phase of the Biomarkers of Aging Challenge. And I'm going to present this on behalf of Team Kauai. Team Kauai consists of my brother, Träuble, who's a PhD student at the Helmholtz Institute in Munich, of Raphael Lerma, who's a PhD student at the German Institute for Cardiology, also in Munich, and of myself, I'm currently a PhD student at the University of Cambridge. So the goal of the second phase was to predict mortality based on either using proteomics data or epigenetic data. And we used proteomics data from the UK Biobank. So the UK Biobank uses the Olink platform, which is a proximity extension assay that can cover up to 3,000 proteins. And crucially, it also contains follow-up data. So we started by first cleaning the data, both on the protein and on a sample level. That meant discarding any proteins or samples which had high amounts of missing values. And for the remaining missing values, we imputed them by the median value. Then we selected the features. First, looking at which proteins are also covered by the challenge assay, because the challenge assay used another proteomics assay. And second, we removed any highly correlated proteins because we thought they won't add any useful information. So these steps led to a training corpus of around 50,000 samples and cover up to a bit over 200 proteins. Now, in order to predict mortality, we first employed a distribution matching normalization step. So as I already mentioned, the challenge assay was different to the training assay. So we needed to make sure that we align the challenge distribution, the challenge assay distribution to the training assay distribution. And we did that on each protein separately using the formula seen here. So just for an example protein, AGER, I think, you can see the distribution of measured protein values in the training assay in red before the normalization and in yellow after normalization compared to the one measured using the other platform in the training set in blue. Then using these normalized protein values, we then predicted risk scores. So for this, we used a survival model. In our case, we opted to use a Cox model, and they are able to fit or predict risk scores by using the follow-up data. So the follow-up data contains information whether the person is still alive or has died, and then also the time point of the follow-up. And then we fitted this Cox model on our training set and then employed it on the challenge set. All right, then these risk scores are transformed into mortality ages or age range again. And for this, we use the so-called Gompertz distribution. So the Gompertz distribution just has similar characteristics of what you would expect of a lifespan distribution. And then these mortality ages were evaluated on a left-out unseen data set from the challenge, and scored according to the concordance index or C index. And just for now, the higher the C index, the better, or the closer to 1 it is. And our approach could yield a C index of around 0.84, whereas the best existing mortality prediction model before this challenge led to a C index of 0.82, and I believe they used epigenetic data. And as we used proteomics data and also a survival model instead of a huge deep learning model, we can also now make this a bit interpretable. So for here, I just plotted the top 10 most contributing proteins to our model. In blue, you can see the ones which contribute to an increased mortality age. And in red, you can see the ones which contribute to a decreased mortality age. And then we took a quick look into the literature and found that our 3 most positively contributing proteins of the model are actually known mortality-associated proteins. So that also validates our approach in another way. Yeah, thank you very much.
Thank you so much, Jakob, Julian, Lucas, and your teams. Incredible job. Thank you so much. So now I'd like to hand it over to Vadim Gladyshev to tell us where to go eat.